Super excited. Beautiful weather, beautiful new venue, actual people, because the last time we did one of these banquets at noon, it didn't work out so well. So we're back to the evening routine, and I'm super excited about that. I am the president of the Entomological of Washington, Matt Buffington. My president-elect, Dr. Don Weber, will be introducing the speaker. But before that happens, a few kind of notes of my own. So the Society has undergone a massive change with respect to the website and the membership directory. Thank you everyone for being patient and renewing. However, if you have had trouble renewing or have been dragging your feet to renew, all the people involved with this process are actually here. So we can help you help yourself. Let us help you. So if you've had trouble, come see us. We can help get this sorted out. My fellow executive members, is there anything anyone else wants me to announce? Okay. They're checking the Zoom link, I believe, right now. Looks like a lot of stress over there, so I'm going to keep on talking for just a minute. So the annual banquet has been, for at least the last 10 years, been the focus of an entomologist who has surpassed kind of your sort of average career goals. They usually could produce a book of some sort. The book is actually part of the banquet where the speaker brings copies or copies are sold and they can sign it. This is no different. So be prepared for that if at the end of the night you want to get a copy. But it does give me it's always a fun time because of the caliber of the entomologists that we get for the banquet, makes the banquet a really special thing. And I would like to offer my own sage advice for anyone who might be attending an Entomological Society of America meeting while David Wagner is actually at the same meeting. Look very carefully in the schedule when David Wagner is giving a talk and when you're giving a talk. And if those two line up, try to maybe find a way to move your talk. Because I experienced this in St. Louis a few years ago, and the room literally just left right as I was getting up. And I went running to see where everyone was going. And there's this like 400-seat auditorium with about 700 people in it. It was an amazing talk. I'm super excited. Dr. Dave Wagner is here to give us a talk. And I'd like to extend my appreciation to Don Weber, who was able to finagle you to come in. So thank you for making the long drive. With that, am I ready to hand it over to you, Dr. Weber?
Sure.
Okay. Here you go.
Thanks, Matt. Welcome, everybody. This is a new venue for our banquet. I hope you like it. And I very much thank everybody for bringing some great food and drink. And I want to particularly acknowledge Professor Susan Agnolini of Marymount. Stand up. For arranging and hosting and scuttling around and making sure the logistics work. And I also want to acknowledge other members of the executive board of the NSAC of Washington, including Lourdes and Matt, and Alan, others who have done a lot of things behind the scenes. I think are still working behind the scenes to make the Zoom work. Do you have anything you need to? Okay.
We'll get it done.
Okay, we'll get it. Okay. So, and let me just say one thing before I forget, but I may say it afterwards too. If you are here, you should go down and see the guard before you leave, and you get a freebie, I think.
Yeah,
right?
you have to tell them you're with a conference.
With the conference center. Those are the magic words. Okay. Better between you and $11. Okay. I think we are sure that Dave had a somewhat traumatic journey down here from Connecticut. We all know that corridor, it's not friendly driving sometimes, and we really thank Dave for persisting and doing whatever he needed to do to get here. He's here. Dave and I overlapped at UC Berkeley back some time ago when I had more hair. It was longer and it was less gray. And I think it's probably true of you too. He was a left guy. I was kind of a beetle person out in the agricultural world, and he was a left person in the Essex Museum a lot. So we didn't overlap too much, but we had mutual friends and knew one another there. And since he got his PhD at UC Berkeley, he has for some years been professor of ecology and evolutionary biology at UConn, up the road, with core interests in the systematics of the Lepidoptera, and now more and more insect decline, invertebrate conservation. And Dave is a prolific publisher, but also an enormous collaborator. And I was just going to read off a couple of interesting titles. I'm going to start with the more entomological ones. So, The Tale of Two Caterpillars and Reclassification. I gotta get my glasses on. Cerathosia Smith and Cydosia Duncan, Lepidoptera noctuidae. Here's another one, a notodontid novelty. The Roa zethus caterpillars co-opt and optimize their anti-predator weaponry to disarm plant defense. Okay, we're still pretty entomological there. Here's another one in review. First report of the family Micropterigidae, Lepidoptera from Central America with descriptions of 2 genera and 5 new species by Wagner and Davis. Yeah, right there. Both of them right there. Now, here's a couple other ones now. Wait a minute. In order. Importance of transmission line corridors for conservation of native bees and other wildlife. Insect declines in the Anthropocene. Insect decline in the Anthropocene. Death by 1,000 cuts. Save Earth's global observatories. Likens and Wagner letter to science. This is my personal favorite. On being a caterpillar. Structure, function, ecology, and behavior. I love it. And wait a minute. One more. With 69 others, I think. Scientists warning of climate change in insects. So, those are by way of introduction. And are we zoomed?
You're Zoom.
We are zoomed. Okay, let's zoom into Dr. Dave Wagner's talk. Welcome.
Thanks. I'm not certain I'm going to need this. Maybe I'll try without it. Can you hear, Martha? You guys can hear? So, but I'll start using this if I start to trail up. I haven't given this talk before, so we're just going to have to see where it goes, especially the latter third one. This is the first time I've ever seen some of these animals on a bigger screen, and the first time I've talked about a book I've been working on for 13 years on caterpillars of Western North America. So hopefully there'll be time, but I'm going to time myself, so you can get the hook done where. And so you keep track of me too. So after 50 minutes, give me some warning or something like that.
You might want to use the microphone for the people on Zoom.
Oh, okay. Let's see how this goes. Can you hear it? Yeah. Okay. I'll try to use the mic.
I'm good.
So what's going on? Oh, you're
I
gonna
wanna
share the
share
screen?
your screen.
Okay, thanks. Anyway, it's gonna be, I think 3 parts. And so the first part would be just talking about caterpillars and how they can tell us some things about systematics and taxonomy that many people miss because they work primarily with adult insects. And then I do a lot of insect-plant interaction work, and I'm very interested in documenting interaction diversity. So the museum's really great for preserving specimens and getting DNA, but at this point, what's going extinct the fastest or being excavated the fastest is actually the interactions among species. They're being broken every day. And so I would really like to be a part of recording a lot of that, particularly the insect-plant interactions.
Switch over when you are
Switch over in what way?
to the screen.
Okay.
But I wanted them to see your pretty face when you
Oh,
were
yeah,
introducing your
yeah.
dog. All right, Alan, do you see the right screen on mine?
How do I advance the slides?
Oh, let's pull you back.
I don't see any.
My bad, let's pull you back. There you go.
Okay, great. Don't go far away.
Come on, let's see if you're ready.
Okay, so here we go. Let's see what happens. So
Call someone up for whoever.
So I got interested in immatures accidentally. I didn't ever intend to get interested in immatures. I was interested in butterflies and moths and beetles and frogs and lizards and everything else. But as an undergraduate at Colorado State, I took a course in immature insects from Howard Evans, and I thought it was one of the coolest classes I've ever taken. And at that point, my trajectory was to become a plant pathologist, botanist, especially mycologist. I was working with mycorrhizal fungi. But Howard was a very quiet man, but just a brilliant mind. And his course was life-changing for me. And then I went, he actually recommended that I go study with Jerry Powell. I didn't really know if I had a career in entomology. And I asked Howard, and he wasn't sure either. He was very quiet and shy, but on the 3rd time of asking him, he said, well, you could try to go to Berkeley and study with Jerry Powell. And then Jerry was very into immature stages. So he studies Microlepidoptera, as many of you probably know, but one of the best ways to get Microlepidoptera is to find their caterpillars. The caterpillars are often much easier to find. Just think of leaf miners or some of the gall formers and that sort of thing. And so that was great. And I learned a lot about immatures from him. In fact, my very first field trip with Jerry, I went along on his immature insects class to Mount Tam in Marin County. But while I was a student with Jerry, probably the real life changer was Fred Stair, and he gave me the opportunity to contribute a couple chapters to his book. And it was, so all of this, none of it was planned, and but this was probably the thing that really switched me over to being very interested in immatures. It was just, he was throwing me a bone as a grad student, and I'm not certain I did a very good job with either of the chapters that he offered to let me senior author, first author. And that was the Elachistids and the Hepialid ghost moths. For a bunch of reasons that I don't really fully understand, I ended up becoming a caterpillar authority. You know, the Forest Service walked in one day to my lab and wanted to know if I could look at the difference in the caterpillar fauna from sprayed forest. This was actually in Virginia, not far from here in the foothills. So they were going to spray forest with BT, and it's a biological control, so it should be great, right? We're going to use a biological control to control gypsy moth or spongy moth. And all I had to do was rear the caterpillars. I didn't care about the science that much at that point. I was a taxonomist or a phylogeneticist or what have you, studying leaf miners, I guess. But I was very, I was interested in the animals themselves. And so they said they would ship me the caterpillars from sprayed plots and the control plots. And basically they were trying to figure out what was going to happen to the bird food and whether, we all throw BT around thinking, it's a biological, so it's going to be great. And in fact, we found out it had a pretty significant non-target impact on various caterpillars. And so It's a biological, but it's a broad impact biological on Lepidoptera. So I think what they shipped me 12,000 caterpillars in 2 weeks, and I had to rear them all individually. And at that point, according to Jansen, caterpillars are the largest organisms in the New World for which there was no literature. And so we didn't know what they were. We started calling them plumbers' butts and speed racers. There were no names for any of these things. And they were just morphospecies. And we took lots of photographs. And from there, my life and my career took a really abrupt change. And I ended up putting little books and then bigger books. And then ultimately, I guess this book, which is now in its 10th printing, it's won a National Book Award. It's been translated into French. It's actually been covered in a literary journal, which is pretty funny. And then this is probably my most recent larger book. It came out in 2011, I guess. And I worked on that for 10 years, and it treats about 600 species of noctuids. But now I'm off to do Western North America, which we can talk about later. Probably the latest thing that I've done with caterpillars, it's fairly substantial, is this 500-page monograph with Jim Miller, the recently deceased Jim Miller. And what a wonderful opportunity. And I feel very privileged to have worked with Jim. He was an amazing scientist and artist and musician, and just really fun to hang around. Wonderful tropical Lepidoptera. These are the notodontids. I think we had over 200 caterpillars. I think I reared close to 4,000 species at this point in time. I enjoy caterpillars a lot. So I don't know if I'm obsessed, but I'm pretty close if I'm not. So what we're going to do is talk about how these are useful, I guess, in systematics, just in terms of improving classifications and alpha taxonomic matters. And then maybe how to use some of this data, this insect plant data, to answer big questions that really matter. And then if there's time, we'll look at some images. I'm really excited about that. I haven't seen them on the big screen. Some of them I've only seen on a computer screen, or even just fix them up, this evening, getting ready to show you. So there's advantages and disadvantages to working with immature stages. So they're not very well known in many cases, but at least among Holometabula, they're at least more well known than most other orders. They have microscopic characters, even ultrastructure. You have to look at ketotaxy and the crochets on the bottom of the prolegs and mandibles. So they're not easy to work with many times. And they are also pretty time-consuming to collect and especially rear. My kitchen, I can't show you my kitchen. I like the animals close to me so I can see what they do. And I think that's what makes my species accounts interesting. And so I'm not working with animals and alcohol. They're almost always alive when I write my species accounts. And so that puts a certain amount of intimacy that is easy to relate to the species accounts. There's a lot of things that are really advantageous about caterpillars. One of the major points from tonight's talk is they have a different rate of evolution. So natural selection acts on all life stages, and sometimes the caterpillar rate will be phenotypically, what a systematicist is going to look like if they have specimens will be faster or slower in the caterpillars. And so they could really be an important and complementary tool set. They lend themselves to being studied alive because they're slow, like me, and especially the older I get, the more thankful I'm studying caterpillars. I don't have to chase dragonflies or tiger beetles anymore because I think they could have both outrun me at this point. So they're essentially a really important part of the biology. So I half the time I spend on invertebrate conservation and insect decline matters these days. But if you want to preserve any insect, you really have to know about its immature stages, right? So that's sort of the quintessential part. And if you're not studying the caterpillars, then you don't really or the larvae, and it doesn't matter if it's a fly or hymenopteron or whatever, you're not gonna, this is the longest live stage, this is the trophic stage. And for the most part, the stage we study is, gonads with wings. And, and sometimes it'd be very ephemeral, right? And so a very minor part, really, of the Darwinian fitness for these organisms. And also, certainly the caterpillars are the economically important stage. And maybe important message to take home tonight, and I'm not certain it's true, but I'll tell you it anyway. Caterpillars may transfer more biomass and energy from plants to the rest of the tree of life than any other insects on the planet. Especially if you look at external biomass on trees, on shrubs, it's caterpillars that make baby birds. And it's caterpillars that turn into moths that feed many of the rodents on this planet and all the, over 80% of the bats and that sort of thing. So much of the fabric of terrestrial ecosystems, Jansen calls it hamburger. That hamburger is caterpillar. So I'm just going to share a couple examples of where larval systematics or caterpillars really tell you a lot. This is a famous paper of discovery of a new family of Lepidoptera from Patagonia, the Heterobathmiidae. And so I'm going to actually skip past this, but Hinton had his system of ketotaxy, and so all these hairs on a caterpillar are named, and that work by Hinton is still used today and was a fundamental part of Lepidopteran classification. And a great amount of information can be had from knowing where those setae are positioned and the shared characters among groups, and also the autoepimorphic features of certain taxa. And this is the slide I should have been talking about when I was talking about these primitive moths, but especially the Heterobathmiidae. And so this was a group of moths. At first, from Patagonia, we didn't necessarily know what the early stages were. Christensen and Nielsen put them into the Mycopterigidae originally, before they had the larval stages. And what they determined, I think they found about 6 different, what they considered synapomorphies, that would put these very ancient archaic moths in with the Mycopterigids as a subfamily. But shortly after they published in 1979, they got the larval stages from Southern Beach or Nothofagus, and they found out that their publication from a couple years previously had to be revised. And so based on adult characters, they got it wrong. And based on larval characters, which are the characters in green here, there were far more characters in green that were arguing that the Heterobathmiids were a separate family of their own, a really important early diverging lineage. And so just 4 years later, they had to recant their original, and these are the top, among the top Lepidopteras in the world, working at that point in time. And so they, based on larval stages, moved them into the Heterobathmiidae. So these are a couple of trees, and this is Jim Miller's work again. And so he had, I guess I don't have a light pointer, but I don't think I really need it. But in these phylogenies, the ones to the left is based on adult characters or adult features. The cladogram on the right is based on larval features. So there was 100 characters that they had. And if you look at the tree on the left, what you're going to see is the 100 adult characters for these notodontids or prominent moths don't have a lot of structure deep in the tree. So if you look at the basal node, there's a large polytomy. So we can't infer too much from the adult characters deep in time with the adult characters. But the other thing that you'll notice here is there's not as much resolution overall in that tree. But with just 63 larval characters, you'll see that there's less polytomies. And there's particularly more structure at the base of the tree. So this was a case where getting larval stages or immature stages were very, very important in terms of the world classification for nododontids. I'm just going to share a couple stories again of how larvae have informed my research and helped me reclassify or understand more about the tree of life for Lepidoptera. So when I first started, again, there were no books that you could use to identify caterpillars. So this is one that I found on the way back from a LEPSOC meeting. This is this green caterpillar, kind of nondescript. I got it. It was very abundant on black gum on the shores of Lake Michigan on the way back to Connecticut. And I took some pictures. And the only thing you could do at that point in time was call other taxonomists or other Lepidopteras. And so it was a funny thing. I sent it to Tim McCabe, and he was sure it was Polygramity hebraicum, the Hebrew moth down in that lower left-hand corner. I showed it to Doug Ferguson here at this USDA Smithsonian, and he was sure it was Comacara cadbury. And they're both Top Guns, and they were both sure, but they were in different families, so they were sure they were both sure of their ID. Unfortunately, I didn't know that these caterpillars would only pupate in dead wood. So I did my, who would thunk it, they all died. And so we never solved the mystery. But then I was interested. So in 2001, we did, I don't know if that was a BioBlitz you went to, Don, but I finally got the life history of Polygramity. And it was really interesting. So this is an early instar, feeding instar. But what's really interesting about this caterpillar that I had refound was that it had 3 phenotypes in the same instar. I didn't know caterpillars could have more than one phenotype in an instar. And so that was a revelation and worth publishing by itself. I still think it's the only one that I know about on the planet. But the feeding instar is green. And then the one in the lower right is a special bark tunneling instar, or not instar, but color phase. So it stops feeding, it turns blue, mottled blue and black, which is great for tunneling on bark for 5 hours because a bird won't see it if it's blue and black spotted. And then once it gets in the cell, it turns red. So I read it all the way through this time with the wood, and I was also starting to get into DNA barcoding, so I knew that this is Polygramity. Then I ran into this other caterpillar that I didn't know, and so I think I have a movie here. Polygramity had this really, really unusual. Let's see if this works. Has a really unusual behavior. So there you can see the caterpillar tunneling how it's that phenotype that it has for about only a day, helps it blend into bark. But watch what happens here. It's going to pull out what I call a soccer ball. So okay. And so this is the first time I'd ever seen anything like that in a caterpillar. And it was a kind of perfectly formed ball. Okay, so if this doesn't advance, let's see, I'll try this. Here it goes, let's see.
Might just need to click through.
Oh, I'm clicking fast. Oh, here we go. So we've been here before. So then it was another LEPSOC meeting when I found Comacara cadbury, and this was the moth that Doug Ferguson said my caterpillar was. Okay. And so I was very interested in getting this and figuring out Doug was always right. And so I was trying to figure out how Doug's train derailed. And so I got a female and I got eggs, and son of a bitch, if they didn't look exactly the same. So,
Wow.
so remember, these are classified in different families at this point in time. So the one on the left is supposed to be a tiger moth, and the one on the right is the Hebrew. And it's actually, at this point in time, I think it's misclassified. They're both misclassified at this point in time. But you can see that not only did the caterpillars look almost identical, but they both had this very special bark tunneling behavior. And so they both form these soccer balls. And I knew when you see a global synapomorphy, you see something that's only evolved once on the planet. It's pretty obvious. And like I said, I've reared thousands of caterpillars, and I knew that this had to be a derivative from a single common shared ancestor. And so it's just a matter of trying to prove it. And it was a little hard given that they were in two separate families. But I went ahead and published this and suggested that both would be dagger moths or in Acronictini. But these guys had befuddled a lot of good taxons. Frankelmann initially had put it in the Saurothrypine noctuides, and then McDonough, who was a great Canadian Lepidopterist, put it in the Archaeidae. Forbes put it back in the Saurothrypine, and then Frankelmann put it back in those guys, and so on. So it was just kind of mess, but the larvae were really what gave it away in this behavior. And so one of the lessons here is that behavior can be, we always think of behavior as being too plastic and too ephemeral, or evolves too quickly to be used in phylogenetic decisions. But certain things actually can be preserved for very long periods of time. Dawn's mycopterigids are probably eating the same kinds of things on, fern spores and liverworts today that they were doing 120 to 150 million years ago. And so that's one aspect of nature and studying live insects or extant species that I think in many cases gives you a window back in time and an exciting possibility. So another one of these moths is this bird dropping moth. It was misclassified as a acronictine, or a bird dropping moth, was Thermus sorrenta. And while I was rearing this caterpillar, when it went to pre-pupal stage, it looked for wood and rolled soccer balls. And so based on that alone, which is kind of silly, I was certain that it was going to be shared a common ancestor with those other two moths. So I was pulling these things from different parts of the tree for Lepidoptera classification and looking for characters. And sure enough, I was able to find characters for that surma. This is another soccer ball roller here that seemed to lump it with Acronictini and the Polygrammidae and the surma, Polygrammidae, and Comacara. So again, there's this ball rolling behavior. There's actually four different genera of moths that were displaying similar behaviors and rolling these soccer balls. And so I actually moved all four of these into a single group, the Acronictini. And I'll show you a movie here of the larva. This is the fourth member. I hope this is going to run. It will
flash through that title that said barking up a new tree.
Yeah, right. Yeah, you missed one. Yeah. So anyway, this is that same behavior again. So I'm
Get it out of here.
pretty sure that this is a global synapomorphy, or pretty damn close to it. But it's really a fun example because the top one again was a tiger moth for the longest time, and two of them ended up in the Acronictini, but the other one was a bird dropping moth. So all of them were brought together basically by their behavior using larval stages. And there's a funny side story to this, and a lesson about barcoding. So I was sure that this behavior was we could go to the bank with it, and that all four of these moths shared a single common ancestor, and I'd moved them all in Acronictini. But Don, I think you were there in the Smokies when this happened with Polly Bear. So when we had Polly Bear come down, it was when he was first getting into barcoding, and he was very interested in coming down to Great Smoky Mountains BioBlitz, where we were going to collect as many species as we could, and then he would pull a leg off and barcode the leg off these moths that were identified by authorities. I think Hodges may have come down for that same blitz, and Doug was there. It was maybe one of the first times Doug and Ron had, and you had all been in the field together at the same time. But Bear was back at, I think we got over 800 species of moths in 2 days in the Smokies, and that became really part of the backbone for the North American database. But what happens is that Harrison Memma, so one of my members, the one I just showed you a video of, the barcode places it in the consanguinity, places it in a completely, it contradicts my belief that it's an acronictine or a dagger moth, and it belongs in the consanguinity. But I was so sure that I was telling Polly Bear that you can't trust your DNA. So so I said, so if the larval characters are and behavior were right, Harrison Memma was an acronictine. So it goes pretty basally in the tree. So so that's where I thought, so Harrison Memma is the middle moth here. And I believe it was related to these other moths, and it belonged in the acronictine. And Bear was and Jeremy DeWard were in the back of the room, and so this is where the DNA, the barcode was putting it. It was putting it near a crown group near the top of the tree. It was always placing it, and to this day, the barcode still places Harrison Memma in the consanguinity. I checked before I came to lecture tonight. And so Bear was convinced that I was wrong because I wasn't using molecules, and that the middle moth would go down to this other group. So Bear at that point bet me a 6-pack of beer. Wait a minute. But he upped the ante while we were there. He says, one day, Wagner, you're going to wake up and ask yourself, how could I have been so stupid? Dave, make our wager a case of beer. Well, he lost.
Get that beer.
So Yad Ronka wrote, one of my PhD students decided to do a phylogeny for the acronictine with Bridget Zakharchenko. So we generated a phylogeny for the acronictine, and the ball rollers are essentially all together. They don't necessarily form a monophyletic group in this tree, but they evolve a single time. And the branch support at this part of the tree is not strong, so it is possible one day that we'll find either all that ball rolling evolved only a single time. Well, so there's two, you'll see that the green ones and the purple ones here are both ball rollers, but we actually don't know what the states are for the related ones. They're from Asia or Africa and other places, so we don't know what the behavior is going to say, but it's either a paraphyletic or a monophyletic assemblage, and that behavior, we still believe that behavior is only evolved once. So anyway, it's a fun story about how caterpillars and their behavior can tell you about things that happened a long time ago and could be windows to the past, and offer us a special opportunity to get a different character set. But it was certainly it was the larval behaviors that tipped me off that these 4 disparate moths belonged in a single clade. So I also want to talk about, so that was a way of using larvae to look back in time and infer something very ancient. I want to flip that around and talk about how we can use caterpillars to show us about recent evolutionary events. They can evolve a lot faster. So one of the reasons caterpillars are going to evolve a lot faster is because they're one of the few insects that are feeding externally on leaves and under constant vigilance from birds. So that's a tremendous selective pressure by itself. And remember, the larvae are sort of the longer live stage. So on average, it's about 3 to 4 weeks for many of these caterpillars where they're under selection for their phenotypes. And so we're talking about situations where there's an up-tempo in the rate of phenotypic evolution because they're caterpillars, because they feed externally. My field guide really only treats external. The field guide works for external species. Nearly every North American caterpillar that feeds externally can be told the species from its caterpillar. Go to the internal borers, the root feeders, the stem borers, or the subterranean feeders, field guides stinks, don't buy it. It doesn't work for those tactics. If you like to go out at night and only collect caterpillars that are on foliage by day, it's a worthless book. So these are acronicta. So I've been talking about dagger moths or acronictines. These are those other four genera showed up in the acronictines. But the main genus is Acronicta in North America, and there's over 80 species. So this is a tremendously successful group of insects. So the average moth genus has about 7 to 10 species, and the microgenera are larger. There's about 10 species per genus of microleps, but for macroleps, it might be 5 to 7 is the average number. And here you have a genus with 80 species in one genus. So they're doing something right. And I think one of the things they're doing right are the caterpillars. Now, if you've seen one acronicta, you've seen them all. They're not properly curated in the Smithsonian, at least the oak feeders. And they're not properly curated in any two collections. Any two collections will have the oak feeding acronicta sorted differently. There's no way to align them. The adult phenotypes are just too confusing. So it's going to require molecular data. But let me take you on a visual journey of their larvae. So this is all just one genus, and this will be fun. So these are all these. So you saw what the adults look like. These are all acronicta. These are all acronicta. These are all eastern acronicta. So you have these within like a mile, most of these within a mile of your home. These are all acronicta. These are all acronicta. These are all acronicta. So they're really phenotypically evolving super fast and super fun, and that's why the field guide works. And they which is really, you don't hear about mimicry. You always talk about mimicry in adults, but there's actually mimicry going on in larvae, which is pretty cool. And it hasn't been written up once. Mayberbaum's done a short paper, but these would be a wonderful subject. So the acronicta are going to be on the left and the models are going to be on the right. So around here, even in the DC area, so you've got some that mimic the tussock moths on the top row and anodontid on the bottom row. Here you have the acronicta mimicking a tent caterpillar, or Lasiocampid. Down below, you have one mimicking an archaeid. So it's pretty amazing. And they can mimic megalopygids, they mimic lemicodids. Now we can use caterpillars to discover species, and this is a really good example. So I had a student, many of you know Tanner Matson. He's on a fellowship on Smithsonian. He's on he just graduated, he had a pre-doc, and he just got his PhD last month. Just had a baby 2 days ago, and he hopefully, Scott, I don't know what this status is with his postdoc. Soon. So it's likely Tanner's going to have a postdoc at the Smithsonian. I won't share the details. So I don't even know some of the details, but there's some details that are worth knowing. But he worked on lectures. He's a small group. They're related to lemicodids. I don't know that we get any species around here, but they're enormously confusing. So when he started, and Hebner was an expert on this, everything, the literature was screwed up, the databases were screwed up. We found out that bold, the records in bold, over 60% of the moths, that's supposed to be the reference database, were misidentified. So dozens and dozens of misidentifications. And really, there was a force. If you look in that, I guess that if you look here, this is part of the barcode tree from bold. You can see that there's almost no differentiation here. So this is, if this were any other group of moths, this would be a single species. But you can see that what's supposed to be a single species based on CO1 or the barcode includes Basostriga, Semitis, Pupilla, and Suburban. So it was just a mess. but I'd been and it had really stumped a lot of really good people. So Harrison Dyer and McDonough again, August Buss and Hodges, everybody was having trouble. The genitalia wasn't helping getting it right. The adults weren't sorted properly here, and the molecular vouchers were all screwed up. So the Rosetta Stone, this is again just talking about how bad Bold was. So more than half of their specimens, or about half of the specimens, were misidentified according to us. And the Rosetta Stone was the larvae. The larvae were all very different, and they weren't variable within species. And so those 4 species immediately sort out if you have the larvae. And so then if you sort these things out and run the barcodes, the larvae and the barcodes make perfect sense. And so that was a way that we were able to use larvae to straighten up the taxonomy. This is another example. I really like Symphistus. It may be the largest genus of Macrolepidoptera in North America. You probably don't know much about it. There's only 4 or 5 species around the east. For some reason, they're exploding. They're radiating very, very quickly in Western North America, probably because of an explosive radiation of penstemons, and that's a story that hasn't been told yet. But the larvae are evolving much faster than the adults. Just an example from the east here. This is actually 3 species of Symphistus forbesi and Cyananthe are shown here, and there's a third that's in this mess. They all look about the same. Don LaFontaine did the genitalic dissections for us. He's a noctuid expert, world-renowned, found no differences between the 2 I'm about to show you. And so there was no adult external characters that we had, and the genitalia didn't work. And we even looked at the DNA, and it didn't suggest anything too obvious. So here in this tree, here's the 2 species. This is the new one we're going to describe, and here's the one from ash that's found around here. But there's almost no difference in the CO1 barcode for these 2 clusters, and it's certainly less than the 2% that we normally look for. But what is going on here is that this is a recently derived entity, and so it does cluster as a separate entity from the ash feeder. And the larva is going to tell us that this is a good species, even though the genitalia and the adult phenotype and the barcodes tell you, but we would not know it without having the larvae. We wouldn't be able to look at this and know that this bottom entity is actually a distinct species. And that's because this 2% that we often use as entomologists to sort of separate or initially separate or start to ask ourselves whether we have 2 taxa. To get 2% divergence in CO1 takes about 1 million years, and certainly anywhere from 500,000 to 1 million years to get that 2% divergence. But we know speciation happens much faster than that. So occasionally it's happening in 25,000 years or something like that. So we know there are cases where the barcode isn't really going to be obvious. Now, if I had huge numbers, if I had 100 of these and 100 of these, and they were all clustering separately, then I would be tipped off that they are acting differently and cohesively. So here are the caterpillars of those 2 animals in that last barcode tree. So the caterpillar on the left is the new species, and it feeds on Triostemum, which is in the Caprifoliaceae related to honeysuckle. And this is the Cyananthe, this is the ash feeder, and very, very different animals. In fact, their behavior is quite different. The Cyananthe, the ash feeder, looks like bark, and it's a commuter. It actually comes out of the leaves. You can tell what a caterpillar does by looking at its color. You can read it. And if you see a caterpillar that's this color, it is not going to be on the leaves by day. So this guy is actually a commuter and can go from leaf litter on up into the tree under cover of darkness. Whereas the new species stays just on the host plant by day. But you can tell that just by looking at the color of a caterpillar. There are some caterpillars where their external features are so closely tied or aligned with their host that you can almost look at the larva and tell what bark. This looks like hackberry, and there's a good chance that it feeds on hackberry. I have some really fun slides. If I can get to them, I have some really fun slides of camouflage, and where we can look at some pretty amazing pictures. And for those of you sitting close to the screen, you might want to move. If we get to the last third of the talk, which I'm going to rip through, you might want to move your chair farther back. But anyway, just in terms of larval features, in the last 4 years, I've discovered 3 new species of Saturnia, giant silk moth, to the United States, which is pretty phenomenal. I don't think one's been described in quite a while, but 3 new species based on larvae. This is the first one. These are gorgeous animals. This is a Sphinx that I found it's only found in one tiny palm forest in southern Texas. So that's one. And this has been submitted. I found a new Saturniid in San Antonio, right downtown. And so it was being called the rosy maple moth. The rosy maple moth has a caterpillar that looks like the green caterpillar here. This is a rosy maple moth caterpillar from around DC. This is the caterpillar in San Antonio that's feeding on buckeye. And so people had missed it because they were, focusing on adults and not larvae. So this is pretty exciting. And the third species I found, I'm not going to tell you about because that's embargoed till I can get it published. So these caterpillars can tell us about ranks. What rank should we recognize an entity at, taxonomic rank? So this is the monograph that I did with Jim Miller on Nododontidae. And so in this fascicle, all of these moths, they look pretty much the same. We're going to be in the genus Ionassa. In fact, this is an early plate, and you can see all these moths are in the genus Ionassa. The top row, though, doesn't fit much. So these are what Ionassa and great caterpillars. They all feed on oak, and every single species is distinct. This is 6 species of Ionassa, mostly on desert oaks. And here is a caterpillar I found that Jim had in the genus Ionassa, and I begged to differ. So I told him that his taxonomy really wasn't going to hold up, and that we needed to create a new genus for one of these. And it didn't feed on oaks; it fed on Berberis or Mahonia, Oregon grape, or something like that. Agarita in Texas. This is kind of cool. See those little white marks? I don't know what those are, but I think they're fake parasitoid eggs. Okay, so the caterpillars are building in fake signs of parasitism, so that another tachinid mother who would come by, or grab a female, would choose not to lay eggs on a previously parasitized caterpillar. I find these on a lot of things, actually. So they're around here. You have a lima coated that has these. Prochoroides, I don't know common names. Need an 11-year-old kid here. They pop them all off. So putting caterpillars, I'll tell you a little bit about ecology, and then that'll let me get a little bit about my Western guide. So caterpillars are really great organisms because they're kind of slow and easy to study, and now you can identify them. And so we've been looking at host plant specialization. And so one way you could use caterpillars and their diet breadth is to answer the question of whether or not tropical animals are more specialized than temperate animals. And so that's a very important question, because we know the tropics maybe have 80% of all insects, but we don't know why. And so there's lots of hypotheses about why there's more insects in the tropics. It could be because they don't have winters like we do. It could be that they have much higher primary productivity than we do. And it could be because they haven't been glaciated in a long time. So there's actually about 7 or 8 different hypotheses. But one that doesn't get much traction, that I think deserves more traction, is the niches are finer in the tropics. That these organisms get highly specialized over time. And so they perceive the world in a much more fine-grained fashion, and you can have denser species packing if you perceive the world in a fine-grained question. So let's use caterpillars to answer that question. Are the diet breadths of caterpillars in Washington, DC the same as they are in Connecticut? Are they the same as they are in Costa Rica? And then let's go down to the equator and let's compare diet breadths across this latitudinal gradient, because it's a really important, if caterpillars are doing this, it's probably true of birds and it's probably true of frogs. So this is a really important question where we can use caterpillars. And this paper came out in 2006. Scott Miller was on this paper, saying that according to our data, caterpillars in the tropics and the temperate zone have equal diet breadth. And that was a real surprise to me because I live in Connecticut where it starts to get far north, and I see a lot of generals. So, but this study was actually a really good and a very tight study and a lot tighter study than any previous study beforehand. And this is looking at, it was a phylogenetically controlled comparison of diet breadths in New Guinea versus Europe. So Novotny, his lab is in Europe, but he has a huge biological research project in, where in New Guinea, Scott? Medang, you know, so he only picked about 15 taxa to compare. And so he's looking at diet breadth and the tail here off to the, as you move from left to right. So the specialists on the left and the general, as you move out to here, this is something with maybe 14 hosts like that. And they're really pretty similar. So he did not find a statistical difference between the caterpillars in New Guinea and the temperate zone. But we did a larger meta-analysis in Nature that came out in 2007. And our data showed something very different. I'm still not sure why. But I could talk about it afterwards. But we found, so at 60 degrees, which would be the far left, diet breadths were broader. And as you move down to lower latitudes, diet breadths started getting more and more special. And so it's a very important question. The jury's still out. Another paper more recently is in 2015. And I believe Scott is also an author on this paper, as is Novotny and myself. This is my data, the Connecticut data here, and you can see that you get longer tails. So if we move from Ecuador and we move up to say Canada, the tail becomes longer, which means there are more host plants, and so the diets are getting more generalized. So Novotny signed on, Scott signed on. That there appears to be a signal that temperate organisms are more generalized in diet than tropical. And so, big question, but the jury, I guess, is still out. But certainly by the time you get to Canada, it seems like most of the caterpillars eat the same plants. But anyway, we'll figure it out with more data. But I am interested in this idea of, so I do a lot of caterpillar collecting these days. And I'm really interested in these species interactions and trianthropic interactions. And I won't have time to talk about it tonight, but imagine that these red balls are plants and orange balls are caterpillars and yellow balls are parasites. And so these are the webs, and we're trying to figure out whether or not these food webs look different in the temperate zone versus the tropics. And if these things are highly specialized, like in the tropics, we expect this to be more linear. And if as they get more generalized, more costly. And so I'm continuing to collect that data, particularly in desert ecosystems. So I'm going to skip past the summary points because I want to get, I only have 10 minutes left. And I hope I made them clear enough. This is an amazing caterpillar, by the way. This is Harrison Memma. So this is the last ball roller video I showed you. And so it's, that's the one also that I won a case of beer with. But the best part of Harrison Memma is its caterpillar. And it saves its head capsules between molts. Can you see? You guys are going to need to move. The good stuff's coming. So Anyway, I think, that these are actually gonna be mace, it's a mace. That it uses to knock off parasitoids and ants. And it's really helpful. If you can knock off one ant and that ant is a scout, it makes your day. Or parasitoid, right? All it takes is one sting. So if you can knock that off, but especially ants, because ants get knocked to the ground and they have no idea how to get back up to find a caterpillar. So it really pays off. The acronicta will actually turn around, grab an ant with their mandibles and throw it off the plant if they get grabbed. So I have some acknowledgements in here for the talk before the Western group, right? Western book. But anyway, a lot of people have helped out. My grad students have been tremendous and raised a lot of caterpillars and had a lot of help from a lot of people. So caterpillars of Western North America. This is my passion these days. I spent 13 years. I think at this point I have 1,725 species accounts written. I have over 1,500 pages. So I've filled up 3 volumes, and I'm on my 4th. And I think I'll stop at 4. But really, I want to say a little bit about insect decline and climate change because I think it's urgent. So this is my PNAS paper, Death by 1,000 Cuts, and there was 12 different anthropogenic stressors that are wreaking havoc on Mother Nature. And we know we're in a biodiversity crisis, and some of this is nitrogen, some of it's pollution, and some of it's agricultural intensification and what have you. But 5 of the things in this are climate change. It's storm intensity is very, very different, and climate variability and fires are, as you know, you probably got some smoke from Canada last week. Fires are burning in the permafrost right now, and Brazil and California has lost tremendous amounts of acreage. I think what really is really scary for me is this middle bottom one, and it's droughts. And so 1 to 2 degrees centigrade is not much for a bug. They go through 30 to 40 degree swings in centigrade, maybe on a daily basis. But water is the difference. And insects are, I don't think that they are all surface area, no volume. They can't store water. And they're screwed because they have to ventilate every cell group in the body through their spiracles. And so that greatly increases their surface area to volume and the stress that drying out does. And you guys are sheltered here in Washington, D.C. because your hydrology is not changing very much. And in fact, it may actually be increasing. The parts of the world that are drying out right now are under severe climate stress. And millions of people are actually suffering in the Sahel right now and Madagascar because of water. The two most rapidly changing ecosystems for us as entomologists are going to be mountaintops because that snow is leaving. That snow is a lot of albedo, and so it was not only was it blanketing and cooling, but it was reflecting a lot of the solar insolation. And as that melts, you're going to have wet ground, and it's going to dry out. It's going to really compromise those animals. So mountaintops are, especially if the ecology was controlled by snow, they might be changing at a rate 4 times greater than Washington, D.C. or some extraordinary rate. I'm very interested in droughts and what they mean. I think we've already talked about that, but droughts mean so much. They diminish nectar resources. They dry out the soil. That's where all these desert insect diaposing states are. You lose surface waters and lowering of water tables. And so we don't know what's going on below ground. We have such agricultural intensification across the planet, and we're pulling water, sometimes very often fossil waters from aquifers. And we don't know what's happening to the soil moisture levels. We don't see what's happening below water. We don't see what's happening below the soil surface. Less snow cover is really, really serious in many ecosystems, but it certainly leads to less water delivery, elevated fire risk, lower plant fitness, etc. And I think that the droughts are going to drive planetary changes in plant distributions. Plant distributions on this planet are determined by moisture, temperature, and soil type. And if you start changing the hydrology, you're going to change plant distributions across the planet. And surely 1/2 of all the insect life on this planet are specialist herbivores. And maybe another half or close to it are the parasitoids of the specialized herbivores. So when you start changing plant distributions, there's going to be hell to pay. We're looking at the worst drought in 1,200 years for Western North America. So this is based on tree ring data. So this has now been declared the worst drought event, and it was particularly bad in 2021 and 2022. I just got back from a sortie in Nevada. Beautiful, beautiful landscapes without insects. Lake Powell's at its lowest level at this point, and so all the downstream states are going to be affected by this, in the agriculture of the United States. Let me just quickly share this example of how important a drought is. So, you guys know the bogon moth? So this is the bogon moth. I don't know if I have another slide. So this insect used to thought to have a population size of about 5 billion. It used to fill caves in southwestern Australia in an analogous fashion to, if you've heard about moths up in the Scree and Yellowstone that provide food for grizzly bears, like 30% of their annual calories are from those noctuids that are under rock. They're estivating up there. Well, the bogon moth estivates in caves in southeastern Australia. This was so extraordinarily common that it would shut down Parliament. It would shut down, trains would have to go slower. Lighthouses would be ineffective. I mean, it was 3 different indigenous tribes used to come to these caves and spend up to a month and just eat thousands of bogon. Innumerable marsupials were dependent on bogon moths. Huge drought about 5 years ago. In the worst on record in Australia. The bogon moth just got listed on the IUCN. So you went from 5 billion to something that's now threatened with one drought. And drought's kind of a euphemism, right? Because don't droughts have an end? And what about aridification? So I think that's my greatest fear, and that's why I want to work on this Western guide. Is I feel like I'm in an urgent race against climate change to document these animals' journeys and narrate their stories, take portraits, give them names that will be emotive, that will excite, but particularly I want names that will make people lament. So let's just go look at a bunch of these guys. So they're fabulous creatures. So yeah, you should sit somewhere else. And so I'm going to go through. I think I've been talking about 58 minutes or something. So I'm going to just go through a lot of slides fast. So these are the wonderful creatures of our American West. I don't have names for, I have names for all of these, but they're not on the slides. Dragon. I get to make up common names, so I call this one a paddle master. There's a little spankers for probably for parasitoids again. Yeah. This cross-dresser, it whatever flower you put it on, it silks the petals. This is illegal in most states. They're both underage. This is my, from my trip with Elizabeth Colbert in October. This one too. This one too. This was a really rare one. Never, it's only known from 2 specimens in the Smithsonian with a manuscript name, and this is the 3rd individual for the species. False head here. Nudibranchs. Yeah, starbursts there. So, new species we found based on the caterpillar. So it's going to be a beautiful book. I love working on it. And these are just some of my favorites. I love the emeralds. These turn into beautiful green moths. Now this, sometimes it's the ugly duckling story in reverse. These are Caculia. Bob Poole worked on those. He's a great taxonomist, but he didn't look at larvae. They look like they're hand painted and put in a kiln. So these are all the adults look, they're pretty hard to tell apart. They need genitalic dissections normally for a species ID, but the caterpillars are just phenomenal. Those are all Caculia. And here's some camouflage. And not only is it the color, but it's the structure, it's the texture, and oftentimes it's even the smell of the host. This is a neat story. These caterpillars, as they molt and they go through their ontogeny or development, they change in each molt to get like an older and older sunflower. They only feed on sunflowers. Here, the middle instars even have the same stamen, the black stamen arrangement. And this is an older caterpillar. And as they feed, they fill they become discs, disc flowers. So here, so their ontogeny is recapitulating the phylogeny of the phenology. They're mimicking the host from the early stages to the point where the sunflowers are actually dropping off their petals. This is a really great group, Grotellines. No species had been reared of any member of this. These are all in one genus, and now we've reared them all from Western North America. They contort themselves to not look like caterpillars. They look like flowers by day. Those are the adults. This is one species account. Maybe I'll read it. This is what the book. Yeah, I'll read. This is indulgent. I'm sorry, and I'll be done here in less than 5 minutes. But this is an extraordinary story. So McFarland, 1988, discovered an extraordinary aspect of the animal's biology, which has now become a heralded example of phenotypic plasticity. Noel found that spring generation larvae, which you're seeing here, which feed on catkins, they bear a close resemblance to their preferred food in appearance and tend to rest among flowers. But oddly, summer and fall generation caterpillars that feed, this is the summer and fall generation caterpillars that feed on older foliage instead resemble twigs and prefer to rest on such. And so Eric Green in 1989 in an elegant experimental study showed that the 2 forms with their attendant differences in diet, appearance, and demeanor could be induced simply by feeding sibling larvae different concentrations of tannins, phenolic compounds common in the summer foliage of woody plants. And in so doing, provided a textbook example of considerable ecological and evolutionary importance of how an organism's environment can turn on or turn off important developmental pathways. Genes. One would be hard-pressed to find a more literal and ecologically important example of the old saw. You are what you eat. There it is. It's a extraordinary thing. So the next 4 slides, I think, are creatures that no one has ever seen before. These are from my last trip to the West, of species whose early stages had never been found. This is an extraordinarily beautiful animal. I love the pinks and salmons and how they blend. So anyway, this is what I'm trying to do before climate change pushes some of these things into oblivion. This is a very beautiful planet we live on. This is the Carrizo Plains in April this month. And not only is it beautiful, but it's little known. And so this is a shout out to Howard Ensign Evans, who made this point, and it was dormant in me for a long time, but it's not anymore. So I think that's all I have. I'd be happy to answer any questions.
Switch this back.
Yeah,
No, I'm asking for questions.
yeah,
Back to your face. It's my.
Yeah.
No, how the heck you raise 12,000 gallons?
if you could push a shopping cart down the street, you got a job in my lab. I think I hired like 10 or 12 people in less than a week. And some people had to quit. They're having nightmares about caterpillars. And some people just don't like caterpillar frass under their fingernails. I can't explain it. So it was nuts. It was really nuts. It was a lot of fun, but I learned a lot about caterpillars in a short period of time. We also killed a bunch of them. I mean,
Do your best.
well, they were all from oak and blueberry, so it made it really easy. It was a very controlled study. So, and oak is, so another thing you should take home, and maybe you already know this, but oak has the highest ecological function of any plant in North America. So I'm going to publish a paper with Doug Calamy this year. We now have over 1,000 species of caterpillars in the United States, north of Mexico, feed on oaks. So there's nothing that's even close to that. And so Tanner's going to add soft lies. And so we actually think the number with snipid gall wasps and soft lies and caterpillars, it's going to shoot up about 1,200 species of insects. So that's like 1 out of every 15 moths in the entire continent is going to be can feed on oak. They're not all specialists, right? A lot of specialists, but a lot of those most of them are general. But really, really important. And then the plant that has the single, if you like birds, cherry has more caterpillars per leaf than any other plant you could. So if you're going to plant 2 plants in your yard, plant a cherry and plant oak, especially white oak around here. A white oak, yeah.
Hi, Dave. Have you found out if they're hungry, they're willing to go to the home?
If what?
If they're hungry, or are they willing to take they would go?
Yeah, so around here, the buck moth would be a good example, and so they'll only lay on oak, I believe, and then but the late instar larvae will move on to other plants. The last instar, so but for the most part, for the most part, they die. So if you don't have the right host, they just die, and it doesn't seem very adaptive. Any other questions? Yeah, Don.
First, congratulations on some beautiful photographs.
Well, I appreciate that.
Yeah. And I've just always thought, that when it comes to herbivorous groups, they would, grow widespread groups, living with lots of different probable species. And there's always going to be a lot more species other than in tropics than in temperate areas, simply because there are more plants species.
Right.
More hosts. So there was no modification. That always,
So that's a true statement. There are more herbivorous insects in the tropics because they're way more diverse. And so that was the point of Novotny's paper in 2006, I believe it was in Science. And it's so it gets ridiculous by the time you get to Yasuni or Tipitini. So this is on the east slope of the Andes, when you get to the equator. There, in a single hectare of forest, there are plots that have essentially the same number of tree species as all of North America, north of Mexico. Over 700 species of tree species in a single hectare there. And so, of course, you're going to have a super high diversity. And so that's part of it. But we also think, that their niches are finer. And so there's more specialists. And I also think that, so So the part of that hypothesis that I didn't necessarily explain too is that we think that diversity begets diversity. So that once you get to tens of thousands of species, that actually is a better catalyst or a better engine for getting more specialist species. So it's self-propagating, or it's a positive feedback. Any other questions?
There's a question.
So there's a comment in the chat, so. Just the initials here.
One thing I'd like to say is I said that I wanted to take their portraits instead of their pictures. And it's a subtle difference, but I think it's really important, and it gets at what I'm trying to do with these Western animals before they're gone. So I want a picture that conveys something about them or makes them of interest. So there's a big push to put animals on like this white background where they're extracted from their habitat. You know, they're really great for field guides. But to me, those animals are naked. And I think I'd like to see their resting posture, and I'd like to, or see a defense posture, or see their host, or see, there's a lot of phylogenetic information in how an insect perches naturally. You can get at it, but their resting postures, at least in Lepidoptera, are just loaded with phylogenetic addresses. It's like going back to Ferguson. He misidentified my caterpillar, but he had the right phylogenetic address. He was very, very close. Even though it didn't seem like it at the time. Yeah.
What has been that for books?
Yeah, that's new. I just stumbled on this. I guess I knew the West is drying out, right? So and so I've been trying to take as many trips as I can and keep my job at UConn. And now with COVID, I can teach remotely. I can get away with stuff I couldn't get away with before. So and I'm going to until so it was just in September, August that I started realizing that we're going to solve this climate change, and we're going to build a sustainable economy, and we're going to decarbonize in about 35 years, I figure, and we'll get it right. But that's going to be too late for a lot of these creatures. And so I have this imperative now to try to find a lot of these things before I can't anymore, or anybody else can, and then try to write a narrative that actually is engaging. We can write a whole book about almost anybody in this room. We ought to be able to write a really great 500-word essay about every animal that we work with.
So I'm the person who wrote the chat about the biota of North America. It shows, it has maps showing the number of species of plants per county in the US and many other maps. So if you wanted to know how many there were, you could look it up, the number of
So
species.
you'll have to repeat the question.
I can read it.
I don't know.
About the biota of North America. It has maps showing the number of species of plants per county in the US. Because the hotspots are California, the Panhandle of Florida, the Smokies, and also the Mid-Atlantic. So you can look up density gradients and PONAD. Maybe this is useful, and then give a link.
So I guess you're just talking about generally the biodiversity hotspots for the US. So there's a number of species, which is really great. And that tends to be very high in the East, because the East is one Arctic tertiary forest from Wisconsin to Nova Scotia, down to northern Florida, Panhandle of Florida, and then over to the Big Thicket. So that's a big biological island. So you have very high alpha diversity at any one spot. But I'm more interested in beta diversity or endemism, and so when you get into places that, it's the only place on the planet where you find something, and so these sand dunes out west, almost always the big sand dunes all have endemic taxa, and so California has the highest rate of endemism of any state, and so that's a place to start, and then there's a higher per capita rate of endemism on Hawaii. Everything is endemic there, and that's the island is very threatened by invasive species and development. And so there's a lot of work that needs to be done there, but it's also depauperate, very depauperate relative to California or something like that. The place in the United States where I see the greatest number of species, if you want to plan a field trip yourself with take your family and take yourself, but is the Chiricahuas in southeastern Arizona. So it's a sky island. It's biodiversity increases as you move south. So that's the tallest mountain range at the most southern point in the United States. But it is really, really rich. It's the highest butterfly diversity, highest bird diversity, highest reptile diversity, I think. But the sky islands of Arizona are a real treat to go to in good monsoon years. And I encourage anybody and everybody to go to the sky islands, any of them. And they really need to be collected because we're already seeing a diminishment in numbers and kind. Yeah.
Have you done much looking into the morphology of pupa as well?
No, there's a lot of good characters in the pupae, but they are simplified. They aren't under so they're not under the selection that an externally feeding animal would be. So I expect that they have a much slower rate of phenotypic evolution. And so they would be good for inferring deep level phylogeny. But I mean, molecules, we're going to infer phylogeny based on molecules, like it or not. That's what it's not feasible now to infer phylogeny based on morphological characters so much because it takes a lot of time and you need collections. Just getting pupil collections from everything sounds impossible to me. And we are in a race. We are in a biodiversity crisis, and I could be dead wrong about this, but I think you have 30 years to decide what to do with your life. And I would discourage you from doing pupae right now. No, I'm just saying. I think everybody in this room should make decisions, and I'm also saying that everybody in this room should make decisions about how they want to spend the next 30 years of their life and what Mother Nature would want you to do if she were at the table with you tomorrow night at dinner, and you guys had a conversation about what would be a good use of your time. Yeah.
Photographs of the beautiful characters, and I'm a fly person. That's wrong. But can you have yours? Yes.
Oh,
Highlight how to photograph. And secondly, what if you put some like the natural environment, well, the habitat in some of these photos, because that you have to text this. We might be like, wow, no, I think that I can move it.
So my first guide tried to have like the natural host plant in the background, and I look at it now and I don't like it because a lot of the background went black because it has to be, for me, it has to be flash photography. So insect photography, to me, the most beautiful images of insects are going to be done outside, long exposures up to 1 second in the morning with before the sun's come up very high, and you get these really beautiful soft pastels. Okay, that is about and then if you stack your images, that's the best you can do to me aesthetically. But I don't have time for that. I mean, and you have to get up, you get set up and do it outside. And then there's all the image stacking. And so I leave that to the nature photographers or people that so love their animals that they're willing to spend an hour and a half on one photograph. So my charge to myself is actually to work at a very, very high volume. And so I take about somewhere about 7,000 caterpillar images a year. And I don't have time. And I know that that's what I should be doing. So and I think the digital cameras are so good now that you can back up, only fill half frame, and then crop down and get a very, very outstanding National Geographic quality image. And it's just you being with that animal. Now, I do cheat. I do a lot of my stuff in this, everything's studio in the lab. It saves me a lot of time. I can do 7 to 10 species, in a half an hour. And you just can't, with a high-end photography, you can't do that. And so I just have to decide, it's not my job as a systematist or taxonomist to do that. If you're studying 4,000 or 5,000 species, it's not the best use of your time. And I worry about that a lot. And I just think that it's going to be another group of people that take those really, really outstanding, soft light outdoor images. Yeah.
For these Western species, have you been able to get any of the portraits of parasitoids?
I have some great parasitoid stories, and I'm starting to sequence tachinids now, but I'm finding some really exciting stuff. I like the tachinids. I don't like them actually. I mean, they kill my caterpillars, and sometimes it's really the only one I've ever found, and sometimes it's find the stuff in deserts, in timing visits. It's hard sometimes, but it's good data. And a lot of people like tachinids, and so and they're really important caterpillar fillers. So I'm saving and barcoding tachinids right now. And I wish I could do more. I could, if I had more help, I could have all kinds of yellowfrids and other proconids and ectomonids coming in with real, Jansen's data is just phenomenal because he's, he has like 10 to 20 hoops and arrows helping him all the time, and we'll have that tremendous data. But I don't have that kind of. I don't have that bandwidth, and I don't have the kind of money in it.
Is he doing caterpillars too?
What's that?
It's Jansen doing?
Yeah, Jansen
caterpillars?
collects the caterpillar. So Jansen started with the caterpillar, but he rears them individually, which takes up a lot more room. But he has huge numbers of people helping him, but he saves the natural enemies and has tremendous data on the host plants, on the caterpillars, and their natural enemies. And the natural enemies are actually going extinct faster than all these things I'm whining about, right? So as you go up in that trophic pyramid, things erode from the top down. And so specialist hypers, which aren't very many, hypers tend to be more generalized, but they would certainly be disappearing right now at a phenomenal clip. But even the parasites have to be declining more than what you're reading in the literature. So when you're reading in the literature that insects are declining at a rate of somewhere between 1% and 5%, 5% in Germany, maybe 1% in a wildland or something like that. But I can tell you that the parasitoids are not part of that data set, and they're declining even faster. Yeah.
Have you tried working with what's in iNaturalist?
So I'm
pretty impressed.
I love iNaturalist. iNaturalist is what everybody could be doing or should be doing, and so I use iNaturalist all the freaking time to determine ranges, look at phenology. My book, I'm only taking the caterpillar images. I plan to use iNaturalist images that other people have taken of the adults. So I have a live adult, and then I have my caterpillars. I want my book to be a wiki. I can't, my brain is too old now to keep track of more than 1,000 species. And it's already getting close to 2,000. And I can't get it right anymore. So I want this to be a wiki project where there's actually a couple hundred people that are contributing data, and I'm only the puppeteer, so, yeah.
I was curious about the difficulties of keeping a larva collection. You need to compare because some of these guys have such complex patterns. I was curious, do they lose their color?
They lose everything.
So you need a lot.
That's the other reason. I think that's why people started photographing caterpillars in the place. They have very ephemeral colors. The greens are really ephemeral,
They all break down.
really fast. Like you drop a caterpillar in alcohol, within a minute, you're like, it's tragic,
So you need a live collection if you want to do all this.
Well, they used to do is they used to bite off or cut off the butt of the caterpillar and then stick an eyedropper in the butt, clip the butt to the eyedropper, and then they would blow into the eyedropper over a flame. And you'd have it inflated, blown up.
We have a whole drawer for those in the
I gotta go see some caterpillar.
Oh, yeah, and not only that, but some people would even paint them afterwards to be, and, so it's like, taxidermy for caterpillars or something.
Yeah, I do that with my grade.
But, I am starting to pickle most of my caterpillars. Just, so it's pretty, I'm advocating for these poor animals, but then I'm boiling them or, immediately, but there won't be many caterpillars for study. So I'm actually, doing my best to preserve caterpillars, but the photographs are the only
That's way to
way.
get those patterns.
Yeah.
So, mine, I have to not be self-written.
Yes.
Do you feel like your work is inspiring us to read more, carrying it out?
Yeah, in a way, I mean, I hope so. I mean, I teach global change ecology, and I know I inspire my students. The caterpillar book, I don't know if I'm responsible, but there's now a Facebook group with 19,000 people for North American caterpillar identification. And the Caterpillar Lab is a thing. And so I think I hear other entomologists that are now getting into photography. They're easy, they're slow, right? They're really good for insect management. And then and they're really important for invertebrate conservation and how do you manage habitat. What is critical habitat? Well, the critical habitat's what's important to the caterpillars. I mean, that's a really important part. So I don't know. Yes. And so you can all do stuff, right? So fighting climate change is the most important thing. That rewilding just parts of your yard is a really good thing. Eating lower in the food chain, there's but really to change fast enough, everything should be focused on policy. So we can all buy better, electric vehicles, and we can eat lower in the food chain, and we can do whatever we want with our yard. But that's not going to save Mother Nature very much. It's good to be an example and do all of these things. And this is what I teach my students, especially like getting rid of like 10% of their lawn and turning that into a backyard laboratory, a nature observatory. But the only thing that's really going to count is changing policy. So if that's the only thing that would help Mother Nature move the needle fast enough. And so if you decide you want to help fight climate change, you've got to get some skin in the game in terms of making phone calls, writing op-ed pieces, contacting legislators, doing whatever you can, starting petitions, getting local town, but it has to be done through policy, because that gets other people. Passing laws will get I'm preaching to the choir here. And that's not, choir's not big enough. Cool. Thanks.
I'm gonna actually find my book. If anyone else has either purchased or has brought a book they want signed, and I'm also going to present Dave with a brand new copy of the C.B. Riley biographies.
Nice.
He's the founder of our society, the Ancestral Society of Washington, and the establisher of the National Insect Collection, among other things, which you can read about that in detail at your leisure. And
I was at that symposium. You had a symposium about Riley, right? And this resulted from that?
Yeah. No, well, it was associated, followed that actually, followed, but it was, yeah.
Congrats.
And so you need to autograph that, and I need to autograph that for you. And Matt, you were gonna take some sort of picture, weren't you?
Yeah, I wanted to exchanging autographed books.
Okay, yeah, we can stage that later.
Yeah.
Let's let everybody, I talked too long, so let everybody get to the food or the restroom or whatever else.
Food, the restroom is the next aisle over and to the left. And did you remember this part? If you came by car? Okay. Question?
Stamp, or will they give you the sticker?
No, he takes the number down and there's some magic. So,
You left it in the car, you want it.
yeah, I guess. And come up here and have Dave sign your book.
Yeah.